This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV. This Week in Virology, episode 1203, recorded on March 21st, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. It is 49 Fahrenheit, 9 Celsius here, and I'm looking forward to talking about viruses.
Yeah, we are going to talk about influenza viruses and influenza vaccines today. Both papers, similar topic. Here in New York, it is 10 Celsius and sunny-ish. And I have to look at my phone to find this out because it's a little windy, I think, here.
And I'm excited about potentially not having influenza since I've had a respiratory infection recently, and it's unpleasant. So yay for things that involve us being able to combat influenza.
Vaccines do it.
Exactly.
Vaccines are the best solution for preventing viral disease. And if you think otherwise, you're just wrong. Do you like being wrong? Also joining us from Austin, Texas, Rich Condit.
I think some people do like being wrong.
Really?
Yeah, sure, because, it gives you something to, be sort of about, okay? All right. Hi, everybody.
Hello.
It is 75 degrees Fahrenheit here and sunny. I don't know why Dixon isn't here. I don't
No
quite
Dixon.
understand the dynamics. Maybe he'll show up before this is over. And a little breezy, but it's a gorgeous day. It is springtime here, believe it or not, in Austin, Texas. The bluebonnets are blooming, or starting to.
Wednesday night, my live stream, I get a couple of people from Austin, actually.
Oh, cool.
Coming. Some guy, not from Austin, got on and said, in the 50 years the NIH has been in existence, it's done nothing.
Oh, is that right?
Yes, that's what he said.
I didn't know that.
And I said, I didn't click on his comment, but I said, that couldn't be more wrong. And everyone else was outraged, and they started listing things that have been done. I mean, people just want to say stupid shit like that to get to get attention, right?
Yeah, it's crazy. Not only that, but what's disturbing to me is that it is not unlikely that some of that mantra, in fact, is just repeating what they hear on certain news outlets.
I'm realizing how much of it is related to repeating what you've heard and kind of parroting talking points. I heard at some point earlier this week an interview with one of the parents of the child who died of measles.
Mm-hmm.
They had multiple kids who had measles, and they said, "Oh, the measles wasn't that bad for our kids."
Wait a minute.
Right.
A parent whose kid died of measles?
Yes, exactly. And I'm sort of like, it wasn't that bad for your kids? At some point, it feels like it's about parroting a talking point and not actually logically letting the thought go into your brain.
Wow.
Well, in fact, it can be not bad, but it can also be very bad. And
right.
that's why, and because so many kids could be infected without a vaccine, that's why we vaccinate. They don't get it. Oh my God!
Actually, as long as we're here talking about measles, I probably should have done this as a pick, but I didn't, so I'll do it right now. Remember my yoga teacher, whose day job is a epidemiologist with Texas?
Mm-hmm.
Yeah.
They've put her on measles, so she's that's not her normal gig. But she's collecting data and sending it into the CDC and this kind of stuff. And she was the one who alerted me to the fact, though it's in the news, that maybe you've seen this, in Lubbock, some woman in maternity delivered a child, and they discovered afterwards that she had measles.
Yeah, I heard that.
Which means that basically every kid in the pediatric, the maternity, whatever,
Yep.
was exposed. Now, interestingly, I haven't heard anything further on that. I would kind of assume that if there was a wildfire spread of measles through that unit, that we would have heard about it, but maybe we will later, or maybe it didn't happen. Maybe they lucked out.
Well,
but man.
2-week incubation period.
Oh,
so.
okay. Wow, that's a long time.
Yeah, that's
maybe that may be good infection control.
That's we talked about that with Adam Ratner last week. 2-week, and that's what was figured out by Peter Pannon when he was studying the Faroe Island outbreak.
Yeah, Rich, if you haven't read that book, we spoke to the author, Adam Ratner, last time. That book is so good.
Okay.
It's very good. Very, very recommended.
Cool.
Folks, if you like these science discussions, and we're going to go on for some, as we always do, we'd love to have your support. Many people seem to agree that that we are a source of great science information. And so if you'd like these to continue, we need your financial support. You can go to microbe.tv/contribute. There are a lot of ways you can help us. And in the U.S., it's all federal U.S. tax deductible. Okay, ASV 2025 will be in Montreal, Canada, or Montreal, U.S. I don't know.
Montreal.
I was talking with someone.
Be careful, you're going to lose listeners. Too.
Oh, I was looking at the landing of this dragon capsule.
Yeah, we were watching that too.
They were calling it the Gulf of America.
Yeah, of course. Of course.
Oh, my God. That was disgusting. It's not the Gulf of America. One president should not be able to change that.
Yeah, it won't last. The what was really cool was the dolphins greeted that splashdown.
I saw that.
Yeah, so that was cool.
They're, as far as they're concerned, they're in the Gulf of Dolphin. Okay, so that's all good. What amazed me about that whole thing is, the technology is mind-boggling. That they can pull this off. It's absolutely mind-boggling. And it's so collaborative. And then they get this capsule on board, and who climbs out but a couple of Americans and a cosmonaut, okay? I'm channeling Rodney King, right? Why can't we all just get along? It's not impossible. We can do this. Jeez.
And you can be greeted by dolphins.
Yeah.
Which would be like a life goal.
It was pretty cool.
Dolphins are awesome.
I remember in the old days, the capsules used to go in the water, then they'd pop the hatch and the astronaut would crawl out. And I'm thinking,
that's right. In the water.
So many opportunities for screw up.
Oh, they sunk one.
Yeah,
before they decided to float
Grissom's capsule sunk.
Yeah.
And but this seems to be a good alternative to pull it out, get it on firm boat, and then
and this,
the people.
This is the thing, is that nowadays, especially the SpaceX operation, all of the equipment is specialized for this task. I mean, it used to be they just used a helicopter to lift this thing out and stick it on the deck of an aircraft carrier. Now they got a whole boat that's dedicated to recovering the capsule, right?
Yeah, with that special arm in the back, and it must be really strong because that capsule is pretty heavy. It looks.
Yeah, and I had to look up because I wasn't sure that I knew that the boosters were reusable, but the capsules were reusable as well.
Anyway, still speaking of Canada, again on the live stream, we're talking about tariffs, and I don't know how I said. I said, we get this Arctic air from Canada a lot too, because they were talking about cutting off electricity right to New York and many other states. So I said, yeah, you also send us Arctic air, and the guy said, yeah, we're going to tariff that too. Cool.
Well, that's where ASV is this year.
ASV is in Montreal, Canada, an independent nation and always will be. Panama will be independent. By the way, Daniel Griffin's in Panama, and Greenland will always be independent. And so will Ukraine. Screw all those people who think otherwise. McGill University, asv.org/ASV 2025. Not much else to tell you except that the deadlines for ASV Cares and Kitty Core are April 9th. That's if you're bringing kids and you want them taken care of on site. These are dependent care benefits for ASV members. Early bird registration ends on May 12th. ASV.org/ASV2025. I just have 2 very brief news items.
Do you think we can keep them brief?
No, I was just going to say we can talk about this for an hour because it's so ridiculous.
You got 2? I only see 1.
Well, one is just a quote, which I think is brilliant,
okay.
Right?
Fine.
So, New York Times article by Apurva Mandavili. RFK on Fox News says we ought to let the H5N1 infections just kill all the birds and take the ones who survive. They'll be genetically, well, he didn't say genetic, right? They'll be immune and we can use them. Why don't we do that? I don't know who told him this, because I know he didn't think it up himself, right? And this article goes through why this is a really, really bad idea. Now, I was going to ask you guys, did you read the article? If not, I want you to tell me why you think that's a bad idea.
I did not read the article, but I can give you some reasons why I think it's a bad idea.
Go ahead.
Number 1, we need eggs. Number 2, we need chicken and turkey. Number 3, as the virus replicates in more and more birds, it's getting more and more chances to pick up potential mutations. And that, it's getting more chances to roll the dice and get mutations that could allow for more severe viruses to be made. And so if we let the virus replicate more, we're going to increase the chances of a bad mutant coming out. Those are my big 3.
Okay. What about you, Rich?
I'll go with, I'll agree with everything Brianne says. And I will, I'll add to that as far as I can tell, I mean, this is an interesting idea, but there's no guarantee, even close, that it would work out that way. And in the meantime, you're going to lose basically all your chickens. So my guess is that economically it doesn't make any sense either. This is the foul version of the Great Barrington Declaration.
Exactly.
That's exactly what I was thinking.
Great Barrington for birds. Yep.
Yeah, Great Barrington for birds. So it's ridiculous. And this is what you get. It reminds me of injecting bleach.
Mm-hmm.
It's that ridiculous.
It's ridiculous. Also, these infections are typically 100% lethal.
Yeah.
So how are you going to get a resistant bird if it's killing all of them?
A good point.
And they pointed out in the article that these birds are inbred pretty much, and they're all, like one person said, it's all the same bird basically. So there's no genetic variability to latch onto to try and find some resistant birds.
Now I wonder if you could actually engineer a bird to be resistant.
No, that would be interesting.
Of course, it would be a GMO bird then.
It would be a GMO. People wouldn't want to eat it. Yeah, get your own problem. But they also point out that if a farm, right? I mean, culling is what we've done for hundreds of years. And now we have RFK Jr. plus the Department of Agriculture secretary, who's a lady that's a lawyer and a politician. She knows no science. She's saying this is the way to do it, not culling. Where's the data? How do you know that this is the way to do it, Secretary of Agriculture? You have no idea. You're just playing politics.
I think you ought to go ahead and do it. Go for it. Let's see what happens.
Well, the problem is that if you have a herd that has H5N1, even if you have a bird that lived, nobody would want that bird because it comes from a H5N1 infected herd. They want you to kill them all and start over again.
Yeah,
go for it. If all of the chickens on everybody's farm die of this virus, and you have the one remaining chicken, somebody might want it, but
not export. Yeah, export regulations.
Exporters wouldn't want it, but maybe some people would buy your one remaining chicken.
Maybe. I mean, chickens don't live that long anyway.
True.
So you'd have to hurry up and breed.
Actually, have you read? You must have read Cannery Row.
A long time ago, yeah.
Yeah. There's a great scene. Sorry, this is a digression, but you got to hear this. All right. And everybody needs to read Cannery Row. There's a great scene in that where the bums go out to hunt for frogs for Doc. All right. And along the way, they hit a chicken on the road and it's dead. So they decide they're going to camp. They decide to take the chicken and cook it for dinner. They cook the chicken for dinner. They're talking about chickens, and one of the bums says, "How long?" They're talking about the fact they'd killed this chicken, and one of the bums says, "How long do chickens live anyway?" And another one says, "Nobody knows."
That's right. We kill them all. The other thing is, this Secretary of Agriculture is really into biosecurity. She said they're going to do biosecurity on chicken farms, and they're going to prevent the virus from getting in.
Right.
So, I'm not against trying that, but I doubt that it's going to work because, right now a lot of these places are open, I think. Some of them are closed, but they're just thousands of chickens all shoved in there, right? And then you have some people going in and doing stuff. So what are you going to do? Put them in BSL-3 suits? Okay.
Well, I remember I visited a couple of places in England. I forget what the circumstances were, where we had to do stuff like walk through a tray of bleach to get in. I mean, there are practices for this.
Sure.
There are, but I wonder whether, when I've had to do that, it hasn't felt as strict as it might need to be.
Yeah.
For this.
And I'm not sure how does bird flu get around from farm to farm? Do you know? Is it
farmers do it, birds do it.
Or potentially wild birds.
But wild birds can do it, yeah. I mean, they just drop.
I mean, if it's wild birds, then forget it. All right? You're done.
I mean, the thing is now, wild birds, I think most of the chicken farms are open, and so wild birds just drop down.
Right.
Right. You're not going to get them to bleach their feet.
No.
Okay.
I just think that biosecurity in an animal farm is hard.
Yeah.
It's not going to be perfect, and so you're going to have outbreaks.
And my understanding is that the biosecurity is actually even worse, even more lax in the cattle industry. And so one would hope that they would increase biosecurity there as well to try to stop spread of this virus.
Yeah, well, the cows, those barns are open.
Right.
Birds can fly in. That's what happened to infect the cow, I guess, right? Anyway, another stupidity from the head of HHS. Now, he has previously said that, during the Texas measles outbreak, he said, vaccines are a personal choice. And that's always bothered me. I always thought, how can I counter that? I never came up with anything good. And then Amy actually sent me this quote from the Washington Post by Don McNeil, who used to write for the New York Times. He wrote an opinion column. He wrote, Health and Human Services Secretary Robert F. Kennedy Jr. needs to stop saying vaccines are a personal choice. They are not. Speed limits are not a personal choice. Paying taxes are not a personal choice. Sacrifices are what make us a nation instead of 340 million selfish hunter-gatherers. Each of us gives up some personal freedoms so we all stay alive.
Brilliant.
Perfect.
Yep.
Isn't that perfect?
Yep.
Mm-hmm.
Yes,
Brilliant.
You have to give up personal freedoms to stay alive.
If you would like to live in a community, you have to give some things up.
And there are a lot of people who are cowboys and think they can do whatever they want. Go somewhere else. Get out of here, okay? Because the whole point of the U.S. is that you sacrifice so that we all benefit. And now we're seeing the erosion of that because of people like RFK Jr. And it sucks. So use this to say, taxes, speeding, they're all speed limits. They're all kinds of things that you have to do.
Well, it's interesting because a lot of the situation now is people buying into this notion that the federal government is infringing on their personal freedom and stuff. So now we have a situation where some of that regulation or whatever may go away, and they're going to find out what liberation from those restrictions actually means in the long run. Okay, they're going to find out what they get for their sacrifice of personal freedom because it's going to go away.
Yep. This country was built to be free from the British, right? And then the founding people said, we were going to write this Constitution to ensure that it keeps working for a while. And it worked for a long time. But it turns out if you ignore it, the Constitution, it doesn't work anymore. Brilliant,
yeah, it's amazing.
right?
You
Brilliant.
can just say, yeah.
Or any law, you say, the
Forget
judge
it.
says you have to do this. I don't care what the judge says. That's just the most obnoxious thing I've ever heard in my life. This is not the topic of TWiV, I understand. But, talk about freedoms. You have to give up certain things to make things work. Because if everyone's driving as fast as they can, even in Germany, they have speed limits. They may be high, but they have speed limits, right? Isn't that true, Rich?
Yeah, I think so.
You can't go 400 miles an hour.
Yeah,
right?
but I don't travel in the fast lane on the Autobahn. Tell me, I leave that for the people who are in.
That scares the hell out of me. You look in your back mirror, there's a tiny speck, and suddenly it's zooming by you.
Yeah,
I can't do that. No. Okay, so much for that. We have 2 papers about influenza and about influenza vaccines. And the first one is in Nature Medicine. It is mRNA-based seasonal influenza and SARS-CoV-2 multi-component vaccine in healthy adults, a phase 1/2 trial. So this is from Moderna, who paid for this study. Moderna is in financial trouble, by the way. They paid for this. The first author is Amanda K. Rudman Spergel, and the senior author is Christine Shaw. And I think this is my personal opinion based on what I've heard. I don't think we're ever going to see another mRNA vaccine again for viruses. I think we're done. I think there's too much negativity. Multiple states are trying to pass laws to outlaw them because they say they modify your genetic material, which is completely unproven, but it's hard to get around that. Plus, they're very expensive to make, and the government's not going to subsidize it anymore like they did in COVID. I think this was started a while ago, and I don't think you'll ever see a phase 3 of this vaccine, but I thought we would talk about it because it's interesting.
I think that's actually really interesting because earlier this week I was teaching about vaccines to my students, and we were talking about vaccine types, and they assumed that almost all vaccines are mRNA vaccines. The idea that there was something before mRNA vaccines and that there are mRNA vaccines that are not mRNA vaccines boggled their minds.
Wow, that's interesting.
Today.
That's very interesting.
Right.
Huh. Well, anyway, I could be wrong, but that's my take. Anyway, this is an effort to combine influenza and SARS-CoV-2 vaccines because here are the numbers. 2023-24 season in the U.S., influenza was associated with a hospitalization rate of 81 per 100,000. There were 9,713 deaths in the U.S., and COVID-19 was associated with a hospitalization rate of 132 per 100,000 and 44,240 deaths. 44,240 deaths. Really? Omicron is mild? Oh my gosh. Some people are just ridiculous. Sorry. The atmosphere of the world has just made me very, very critical these days. So they say, we're going to try and make a combined vaccine. And here's another interesting fact. During this 23-24 season, vaccination coverage against influenza is 48.5% in people over 6 months of age and 22.5% for COVID. Isn't that just really low, right?
Yeah.
So
it'd be interesting to know what the I'm sure you can find the data, what the influenza vaccination rate has been over time, and whether there's any sort of recent, because I don't think it's something that gets high uptake on a regular basis.
No, I think you're right.
Yeah,
I don't think it's never high.
I think that influenza number is similar to numbers I've seen before. That doesn't seem outlandish. I wonder with these data exactly what they're considering fully vaccinated for COVID, because 22.5 does seem very low. And so that does make me wonder if they're counting, different numbers of boosters or different recencies of boosters only.
Well, they're talking about the last year. I don't think I've had a COVID shot in the last year, but prior to that I had 7, and I had COVID twice. So
Wow.
I'm not feeling like I'm really due for another immunization. Not that, and frankly, I'm not sure what the guidance is now for this. Do you know?
I don't. It
COVID? For
looks like they
COVID?
cite
Yeah, for
the
COVID.
CDC dashboard
Yeah, the
for that
CDC
number.
recommends everyone have a COVID vaccine of
Annually?
over a certain age. Not everybody, right?
Okay.
I think. Maybe I'm wrong. I just don't know. Let's look it up.
Well, I mean, the fact that we're having this discussion and we're uncertain is significant.
Yeah,
because in influenza, I think most people know. Oh yeah, it's flu season. You got to you should get your flu vaccine. I mean, whether they do or not, they're aware of it. Okay, and I'm not sure that that's locked in relative to COVID. I'm not even totally well. Okay, I'm not sure about.
So basically, they say you should get a annual COVID vaccine, but especially if you're 65 or older.
Okay.
Right. Everybody 6 months and up should get COVID vaccinations initially, right?
Right.
But then, as far as from year to year, everyone should get it, and it should be especially 65 and up. But of course, we don't have any recommendations yet for this year. We have the flu recommendations.
This 65 and up makes me feel discriminated against. You know, you're putting me, you're sort of labeling me with some kind of demographic.
Well,
I'm not sure I feel.
But what
good
if they're
about
discriminating against me, that they don't want me to have enough immunity?
that. Good point.
Well, you could have. They said everyone could get a booster, but you don't have. They said for sure 65 and over, and then the rest is a personal choice, I guess. Silly. Anyway, so the idea, WHO says maybe we should give both vaccines at the same time. That would be better. So this paper is about trying to develop it. Lipid nanoparticle mRNA vaccines are nice platforms that you can combine mRNAs for different viruses and give them at once, right?
It's already pretty standard practice to, if you're due for both, to get both at the same time. That is in 2 injections. Okay. But this, and I think I've done that. This combines them into one shot.
Yeah, I got flu, COVID, and RSV, I don't know, a year ago or something, last season, all at the same time, 3 shots. I don't care, but it would be nice to have it in one, right? Hey, so what they do here, they make an mRNA vaccine called mRNA 1283. Okay, so the original one, I don't even remember the name of it now.
I think the original one is 1273.
1273, the number of amino acids in the spike. And then this is different. That was 1273 is the whole spike. 1283 contains mRNA encoding the linked N-terminal domain and the receptor binding domain from the spike. It's not the whole spike anymore. Very interesting.
It's a little weird, since if the numbers make any sense, it's no, probably. I mean, I'm used to it being the number of amino acids. It should be shorter.
And then they have put in this vaccine also a couple of hemagglutinins from different influenza A and B.
My understanding is that they basically put in HAs that represented what would be in one of the, if not the current, or what was current at the time.
Yeah, I think
tetravalent influenza vaccine.
Yeah, I agree. And I think what they did with the numbers, I think that the original mRNA vaccine from Moderna was 1273. Their new one with this change in the antigen is 1283. The influenza is 1010. So they took a 10 and they took an 83 from the 2 vaccines and smushed them together to get 1083.
Okay. Makes sense. I liked the amino acid in 1273, but I guess we're not doing that anymore.
No.
So this is a phase 1/2 trial, which is safety and immunogenicity. We got, here's more discrimination against you, Rich.
Oh.
We got 65 to 79 years old and 18 to 64 years old. 18 to 64 is a huge breadth, right?
Yeah.
Mm-hmm.
And then you get, also they divide people up by whether you got vaccines or not before.
By the way, for the record, despite the joking and stuff, those divisions are a perfectly rational way to look at
Yeah,
this,
that's right.
because in both of these cases, the incidence of complications and mortality is much, much, much higher in people over 65. So that's a perfectly reasonable cutoff and a good justification, actually, for evaluating those two groups separately.
So they have comparators. They're going to compare the immune response of their investigational vaccine with established vaccines like 1273 and seasonal influenza vaccine. They actually do standard dose and high dose as well. So then in July of 2023, they stop and they collect some data. They have 595 participants in the 65 to 79 cohort, 621 in the 18 to 64. And nobody left the study because of a bad event following vaccination. That's good. Okay. A lot of people are saying now that the mRNA vaccines have horrible side effects, and they're just lying. They don't. There's myocarditis, which is pretty rare and treatable.
And in a particular subgroup.
And that's it. That's it. I don't know where people get this stuff from. Well, as we said once before, if you make stuff up, you don't have to think. Anyway, let's see. Influenza vaccination in the previous season was reported for 68% of the participants, and 55% got COVID bivalent vaccine previously. Okay, that's actually the one age group, and the other age group was 42 and 36% take. Okay, so this was safety. This was well tolerated. There were adverse reactions after 1083 vaccination. So also with the other vaccines as well, the 1273 and the flu vaccines. And the most common ones were injection site pain, headache, fatigue, myalgia, arthralgia, and chills. Those were the most frequent side effects, which most of us get when you get vaccinated. This is nothing new here. Those are called solicited adverse reactions. So they ask you, do you get this? Do you get this? Then there are unsolicited ones. They say, do you feel anything else? And so this is within 28 days. The 65 to 79 cohort, 17% of participants reported unsolicited events. And that's in the 1273 group. That's in the high dose influenza group, 17%. It's not even the investigational one. It's high dose influenza, 13% in the 1273, and 6 to 15 across 1083. So they're all pretty comparable. What were the ones for solicited adverse reactions? Are just 65 to 79 years, 1083, 76%. And you may say, oh, that's awfully high. This is fever, injection site pain. Everybody has that. So
yeah,
the numbers are higher.
The unsolicited events, they're basically saying to you, did you have any medical anything happen to you in the past 28 days?
Yeah.
So that they can see statistically if there's something that they weren't expecting. But that means that people are listing a whole bunch of things that might have happened to them in those 28 days that would be unrelated. Like they point out in the text that one of the things that was listed was a urinary tract infection. Doesn't seem particularly likely to have been related to the vaccine, but they're being complete in their reporting here that did happen to somebody during that 28-day period. So you always have to take those data with a little bit of a grain of salt.
Most of these unsolicited were mild, they say, did not lead to study discontinuation. 4 of them had what they call severe adverse events. One in the high-dose flu, increased hepatic enzymes. And that's considered vaccine-related. and then visual impairment, angina pectoris, urinary tract infection, influenza and pneumonia. So some people got influenza and pneumonia. Certainly not caused by the vaccine because they are non-infectious, right? Also, others, thrombocytopenia, they say were asymptomatic and required no treatment. Remember, they're taking bloods from these people and doing a bunch of standard tests so they can see things that happen, okay? Now, this is a small group, right? There's no myocarditis because it's the rate of myocarditis is 1 in tens of thousands, so you're not going to see it here or anything else that would happen. That's just the way it goes, right? You can monitor for that in a larger phase 3 trial and a post-licensure trial. That's the way it works. There's no other way to do it. 9% of participants in the 18 to 64 had unsolicited adverse events who got the standard dose flu vaccine. 17%, 1273, and 9 to 21 in the 1083. So the side effects is nothing unusual as far as I can see here. Now, how about immunogenicity? This is what we're interested in, right? So they look for antibodies in the serum of these people that will do influenza HA inhibition. So the HA is the target of the antibodies that you're making. And so you can do what's called an hemagglutination inhibition test to measure those antibodies. It's not infectivity, it's just blocking attachment to red blood cells. It's very easy to do. They all made very nice levels of hemagglutinating in HAI antibodies. Similar, 1083 was similar or higher, this is on day 29, to the other vaccines. What was interesting is that I find on day 29, seroconversion rates were less than 40% across all vaccine and age groups for influenza. So less than 40% of people seroconverted. It seems low, doesn't it?
It does seem low. I mean, is it maybe 29 days is not late enough?
Yeah, I think it's plenty late, right?
I don't know.
That brings up a good point, is that we don't know how long this lasts. I mean, this is one of the criticisms of mRNA vaccines. They may not be durable, but you can't tell on a 30-day study, of course. All right, so that's flu. SARS-CoV-2, same thing. High. Here they're looking at neutralizing antibodies because there's no hemagglutination inhibition test. And they get high neutralizing titers against SARS-CoV-2. The ancestral is what they put in the vaccine, the original SARS-CoV-2, and even Omicron BA.4/5. So the neutralizing antibodies are similar across from their investigational vaccine compared to the established vaccines. And again, the day 29 seroresponse rates here range from 21% to 68% for ancestral SARS-CoV-2 and from 44 to 77 for Omicron. So again, it's a little higher than flu.
Mm-hmm.
I'm okay with that. Everybody's going to be slightly different. So basically, this vaccine 1083 has an acceptable safety profile, and the antibody response for flu and SARS-CoV-2 matches the other vaccines that we're using. And they say this supports the further clinical development of 1083, which would be a phase 3, which I'm just predicting it's not going to happen. It's really expensive, and I don't know if there's enough COVID in one place to be able to do a phase 3 trial.
Yeah, you'd have to use an endpoint that wasn't necessarily protection from disease. You'd have to use an immunologic endpoint.
Yeah. So, but also I think, as I said, I think it's not going to happen. It's too expensive. The government is not going to subsidize this anymore. I mean, as far as I remember, they're taking away NIH grants that have to do with mRNA vaccines.
There's been that suggestion. There was a thing going around for a while of a program officer or somebody inside the NIH discouraging investigators in a program from, I think, submitting grants that had mRNA in them because they might get targeted. And I don't really know what's become of that. I wonder about, making a flu vaccine. There are advantages that they cite in this paper of the mRNA platform because you don't need eggs. Of course, you don't need eggs with a cell-based approach either. It's quick. You don't have to worry about modifications of the virus that would be grown on eggs, and it's quick. So there's a lot of advantages. I'd like to see sort of an economic analysis of from start to finish per dose how much it would cost to produce an mRNA vaccine versus the standard egg-based vaccine versus the cell-based vaccine. The other thing I put in here, because we're ranting about how there's no evidence for side effect, downside, danger from mRNA vaccines, and just as sort of some insight into this, and because people are going to come back with this. There was a New York Times op-ed recently that I put the link in the show notes here from Akiko Iwasaki at Yale, a reputable researcher in this regard. It's a very small study that's not yet published. It's in bioarchives. I'll also put that link in as well. Trying to figure out what the situation was with what people called a PVS or post-vaccination syndrome. There are a group of people who feel like let's put it this way, coincident with their they had a serious reaction to the vaccination. And this I'm sure people have heard of, with some of the mRNA vaccinations being, seriously wiped out after the vaccination. And there are those, some in my acquaintance, for whom that persisted for a period of time. And I believe that those people get self-identified as experiencing post-vaccination syndrome. And this is a study by Akiko looking into a group of these people. It's a small group, it's less than 50, trying to, doing an immunological and various analyses to see if there's anything unusual in their profile that makes any kind of sense. And it, reports a bunch of data on that and says, we don't have enough money to move forward with this.
She just looked at people who got mRNA vaccines?
I believe that this is mRNA vaccines exclusively, though I'm not sure of that, and that's a good point. Okay,
it'd be interesting to look at Novavax.
Exactly.
Right?
Yep.
Exactly.
Yeah.
I'm not sure about the answer to that, but if it's not, it should be applied to other vaccines as well.
Yeah, that's another reason why people don't like mRNA vaccines anymore. It causes long COVID, right?
Well, what's going to happen is another circumstance where all of a sudden you need a vaccine right away. And some of that concern, it's all risk benefit, right?
Yeah.
Some of that stuff's going to go away.
No, the mRNA vaccine served us well. If you think otherwise, you're wrong. They saved a lot of lives. They were out in a year. It's incredible.
Amazing.
No, these mRNA vaccines are sort of a wonder of modern science and should get tons and tons of credit. And, I would personally love to see a lot more work done to make them better.
Yeah.
And maybe to diminish any potential side effects, even if whether you say there are too many or not, sure, we can improve. But I think these vaccines are spectacular.
I mean, I don't understand this outlawing movement of vaccines. It has benefited most people. I don't get it. Anyway, we have another paper, Nature Medicine, mRNA-based. Oh, that's the wrong. It's the same paper we just did. Sorry. This is Cell Immunity. Cell Press Immunity. Early influenza virus exposure shapes the B cell response to influenza vaccination in individuals 50 years later. This comes from the Vaccine Research Center at NIH, who's I heard that its future is uncertain, along with NIAID, right? NIAID's being broken up, and this is part of NIAID, which just seems silly to me for kind of revenge reasons, but so be it. This is also about influenza virus. There's this idea of imprinting or original antigenic sin. I like imprinting better.
Yeah, I like
Yeah.
imprinting too.
The other one is too religious for me.
Right.
So kids are typically infected in the first 2 or 3 years of life, and then that whatever you're infected, influenza virus you're infected with influences your response for the rest of your life, and that's called imprinting. And we don't understand it, but presumably involves memory B and T cells, right?
Yeah.
And then you have, sorry, go ahead.
No, I was going to say it presumably involves memory B and T cells. And the data that I've sort of seen about it is that it has something to do with sort of how similar or not similar
Exactly.
different antigens are. And I think that that's, to me, the part that I have the hardest time understanding is it seems like there's a similar enough and not similar enough line.
Right, exactly right.
That we don't really
Yep.
get, and that you can see in work from like Gabriel Victora's lab. But we definitely know that this idea of imprinting and your first infection really is true in flu. I know there was sort of that idea previously with, was it H5N1 versus H7N9? There was like a group 1 and group 2 type of influenza, and it made you more or less susceptible to the other.
Yeah.
So they do mention in this that there's some indication that maybe the same thing goes on with SARS.
Yeah.
So I want to relate my understanding of the importance of this, and you can comment and see if I've got it right or not. Because an issue with flu in particular is this antigenic variation that happens, where there's a drift in the sequence of the antigenic sites in the hemagglutinin that make it so that year after year, new strains can come up that you're no longer immune to, okay? And so we create vaccines and vaccinate against them. But if the changes are sufficiently subtle, if the changes are sufficient such that you may get it again, but sufficiently subtle, riffing on Brienne's point, as I understand it, it could be that this imprinting might make it so the new vaccine to the new thing really has the effect mostly of re-stimulating your original response rather than giving you immunity to the new guy, right?
Right. Or stimulating, as you say, stimulating the original response, because perhaps that response is close enough to the new guy.
Well, that's the big question.
Right.
Okay. Is it really close enough? Does your immune system know what close enough is? Probably.
I think it can figure it out.
Yeah. But I think this paper is trying to address that kind of stuff, right?
Yeah, it does. And historically, it's been hard to study because people are infected with different influenza viruses every year, right? And so you can't get a clean study group where you've never been infected or whatever. But that's what they did here.
Yeah,
and they're very clever.
And the other thing that's sort of nice about this is that you could imagine, does that, how long are you stuck with whatever that previous infection was?
Yes.
Is that going to mess with you for 2 years, or is that going to mess with you for 100 years or for, and so how long is it before perhaps things get quote unquote reset?
Which is an interesting question in itself because, how long is immunological memory generally? All right. And does it actually, my own question as well is, does that vary from pathogen to pathogen? We discussed this a few TWiVs ago. The longevity of an immune response relative to the pathogen that you're exposed to.
Yeah, so according to this paper, it's 50 years because that's the age of the people involved, but could be longer. What they do is they use an influenza H2N2, which first emerged in 1957 and caused the pandemic. So it replaced H1N1, which had been around since 1918. And then it was replaced just 10, 11 years later by H3N2, and it no longer circulated after 1968. So you got an 11-year window of circulation. So you can find people that were exposed to it or not, right? That's very cool. And so they took advantage of this cohorts of people. And this was actually a vaccine trial done in 2018. And they have presumably specimens preserved, which they used to do this study. And in this 2018 trial, it was a phase 1 clinical trial. It was just a safety trial. They got a H2HA DNA plasmid vaccine, or an H2 ferritin nanoparticle vaccine. That would be your prime, and then followed by an HA ferritin nanoparticle boost. Okay, so you get DNA or protein, and in both cases, a protein boost. Okay, is that right?
Yep.
Yeah, that's right.
Right, and then they can study these people. They have people that were never saw H2, and people that did see it. So you can vaccinate both groups, and you can see what happens. You can see the antibodies produced. You could look at the B cells, the memory B cells, as they did here. And they say imprinting is lifelong. That's what they say. 50 years, right? I guess that's close to lifelong. So what do we do? Phase 1 clinical trial. I just told you the design. They enrolled people who were born before 1967. And so these people were exposed to H2N2, and people born after 1970. And so they're naive to H2N2 because 2 stopped circulating in 1968. And so these people were put in this trial getting a DNA or a protein-based H2HA vaccine. So by the way, this H2N2, that could be the next pandemic because it hasn't circulated for a long time, right? Since 1968. Very few people have immunity except those who were living in that time, right? They could be in a good shape.
And the 16 people in this trial who were born after that and got this vaccine.
Got this vaccine, that's right.
So I'm probably imprinted with H1N1. Born in 48.
Yes, you got H1N1 first.
When were you born, Brianne?
79.
So
she never saw H2.
You've either got H3N2 or H1N1.
Oh, wait, 79?
79.
Yeah.
Yes, H3 was circulating still, and then H1 was introduced in 1977.
This graphic for this paper is pretty cool. I'm referring to that all the time. Okay.
So they take this cohort and they say, what's the frequency of memory B cells that recognize? So they looked for H1 alone, right? Specific antibodies to H1 hemagglutinin, and the reason is that there's some epitopes conserved between H1 and H2, and they want to be able to look at it. Then H2 alone, or those epitopes that are conserved between the two of them. That's called H1H2 or H2H1, and so. Before vaccination, the frequency of these memory B cells are the same in the 2 age cohorts. The frequency of MB cells that bind H2 alone was almost undetectable in the H2 naive individuals, which you would expect, right? Very few. They're going to be there, but they're going to be very few. But they could pick up H2, H1, cross-reactive cells in H2 exposed individuals. Then if you immunize with protein, you get a 10-fold expansion of H2 specific memory B cells in these people who have already seen H2 virus, right? But H2 naive individuals get a weak expansion of H2 specific memory B cells after 1 dose. And then after the second dose, you get a nice expansion. But that first one, very little because they've never seen H2 before, right? So the people who have seen H2, they make a great memory response, and the others you have to do a boost to do that. So they say that's expected. Those are the expected patterns that we see.
That is entirely expected with the immunology textbook.
Yes. So this HA protein vaccine, this ferritin nanoparticle vaccine, gives you a very strong H2 specific memory B cell response after 1 dose in H2 exposed individuals. So they're imprinted with it because they've already been exposed, right? So they have a memory to it. I would just say memory, because we don't know about imprinting yet. And after 2 doses in the naive people, you need that second dose to give you a nice equivalent memory B cell response. Okay, next, the antibody repertoire of these B cells, these memory B cells in the different vaccine cohorts. And here they move to modern technology. They sort all the B cells that bind H2 hemagglutinin from 5 people in each vaccine group at multiple time points, and they do single-cell RNA sequencing and amplify the immunoglobulin genes, and they can sequence them and look at them. And later they're going to actually make some antibodies from these as well. That's modern science. That's modern immunology.
Unbelievable.
It's so cool that you can do this, right? So what's the isotype that is made by these H2 memory B cells? It's like IgG or IgM or IgA, right? It's a nice, even I know that.
Good.
Remember, both groups have pre-existing memory B cells, but the ones who are exposed have more of them.
They both have pre-existing B cells, but the ones who have, we don't know if they're memory B cells or not. We know that they both had pre-existing B cells.
Yeah, they bound H2. Right?
Yeah, they have specific B cells that bind, but we don't know if they're memory B cells or naive.
Okay. So most of them, the major antibody produced is IgG by the cross-reactive B cells in H2 exposed or H2 naive individuals. All IgG. I mean, it's 15% IgM and 10% IgA. And vaccinating doesn't really change that ratio. In H2 exposed individuals who are vaccinated, there's more IgA than in the other group that I've just told you. It goes up to 30%. And then the H2 specific memory B cells in these H2 naive individuals were 60% IgM and 40% IgA.
Yeah. So as I look at this, I see that in the naive population, we've got a substantial group of IgM antibodies and some IgGs.
Right.
And in the exposed individuals, we have not so much IgM, a good amount of IgG, and a good amount of IgA.
Brianne, you better
Yes.
review for the listeners IgM, IgG,
Yep.
IgA, because this, has significance, right?
Yes. So these are the different constant regions that are on the antibody, and antibodies can be made with 5 different protein constant regions, M, D, G, E, A. And all antibodies start out, or all B cells start making their antibodies as IgM.
So the presence of IgM is indicative of a recent exposure.
Yes, that's indicative of more of a sort of earlier type of exposure, more of a naive cell that's been exposed the first time instead of a memory cell that's been exposed later on.
What they call a neoantigen response.
Yes.
Cool.
Or if you're a old school immunologist like me, what you call a primary response.
Yeah, that's fine. Yeah. Okay, so the IgM is totally expected for these people that were never exposed.
Yeah, and
before.
so this sort of makes it saying that, okay, both groups had some of these B cells before we gave them the vaccine, but those pre-existing B cells in the naive individuals were more likely naive, and the pre-existing B cells in the previously infected individuals were more likely memory.
Right.
Because they made different antibodies upon that exposure.
And do IgG and IgA as well, because that's significant.
Yeah, so IgG and IgA are things that a B cell only makes a bit later in its life. So the B cell has to sort of switch from making IgM to IgG or IgA. And so what I notice here is that the M production goes away. So we aren't seeing kind of first exposures. we're really only seeing sort of those later exposures.
And IgG is circulating antibody. IgA is mucosal antibody.
IgA, there are mucosal and circulating versions. I usually think about it as mucosal, but Steph would correct me, so I'm trying to be good for Steph here.
Okay. And so part of the significance there is that it is a respiratory pathogen.
Yes.
So you would expect exposure at the mucosa. On the other hand, the vaccine is injected.
Yeah.
Okay.
Yep,
So you would
absolutely.
expect maybe a serum response, and it's always been a mystery to me how you get appropriate immunity even at the mucosa with a vaccine that you've injected.
Well, IgG can get into the mucosa
Right.
too. So, but obviously, if you're infected, you're going to be generating antibodies in the mucosa. All right. So next, they look at the percent mutation of the variable heavy chain. So I presume this reflects somatic hypermutation.
It does. And again, somatic hypermutation is a thing that you see later on in a B cell's life. So if
Right.
there's little somatic hypermutation, you probably had naive cells. And if there's more somatic hypermutation, you probably had a memory cell.
All right. So they can do that by looking at the sequences of the antibody VH chain. And so that they find is that for IgG or IgA, the H2 specific memory B cells in people who were previously exposed before vaccination are high, 6.5% mutations they find. And after vaccination, that doesn't go up. So
they're done.
They're done. They're
Exactly.
done hypermutating. Yeah.
We graduated from from B school. Okay.
Yeah.
We're done.
So then they have antibodies from H2H1. Remember the cross-reactive ones in H2 naive individuals. They had pretty high percent mutation. Remember those are present before as well. But the mean percent mutation of the H2 specific memory B cells in H2 naive individuals before vaccination is low, right? Because they haven't had this opportunity. I'm sorry, it is low even after vaccination. It's 2% early after vaccination, then goes up to 4.5% as the process kicks in.
But, that process takes a long time, right, Brianne?
Exactly.
That can go
Yeah,
on forever.
exactly. So they may
Well,
not
not
have
forever.
waited long enough. It might go on forever. It goes on for a long time, and so they may not have waited long enough for that training to have completed in these individuals.
So I want just to ask you on the side here, Brianne. So you get a polio infection. Could you be doing somatic hypermutation your whole life?
So if you had asked me that a couple of years ago, I would have said no. But I know that some of the data from Gabriel Victoria's lab has shown that the answer is that somatic hypermutation is going on a lot longer than we previously thought. I don't know that we have an upper limit, but I think that they've shown it at least for out to almost like a year.
Are you waffling? Are you changing your mind?
No, she's going with the data.
Oh, okay.
So that means you have to have antigen around that because that's how hypermutation works.
Potentially, yeah.
But the antigen can stick around for a long time, right?
Yeah,
a remarkably long time. This blows my mind.
It could be that, not necessarily say replicating virus.
Right,
it could be viral proteins that are on the surface of follicular dendritic cells.
Right, so in lymph nodes, so it's not floating all around. Yeah.
Right.
Okay. Yeah, somebody asked me.
The longevity of this stuff boggles the mind.
There's some papers that have come out sort of in the past 5 years about this that are just stunning in answering some of those longevity questions.
Okay, so we have hypermutation occurring. So they want to know if actually the antibodies are getting better, right? Affinity maturation. So they use a computer program to look at this, and they can look at the sequence evolution and say, yeah, this is making a better antibody. And so what they find is that the protein HA vaccination expands the pre-existing antibody, both IgG and IgA, H2 specific memory B cells in H2 exposed individuals, again, ones who are imprinted, with no change in isotype usage or somatic hypermutation, whereas vaccination in H2 naive recruits naive B cells that undergo isotype switching and increases in somatic hypermutation, right?
Yeah.
Which we just said. They look also at the clonal diversity and IG lineages over time, and they find that the protein H2 vaccine primarily expands pre-existing H2 specific memory B cells in H2 exposed individuals. Again, right, they have the memory B cells with little evidence for further maturation or a de novo response. So no de novo response, you're not activating any new B cells, right? That's what that would mean.
Yeah, exactly. So the idea here is that we're not sort of seeing a fresh new response. We're just reusing the old one.
And with the H2 naive people, you get a de novo response and a highly diverse antibody repertoire, which is interesting. It is more clonally expanded when you use the DNA as a vaccine than the protein vaccine. I didn't go through the data here, but the DNA vaccine seems to prime. That's the prime. You get a more clonally diverse antibody repertoire. Okay, now we're going to talk about public clonotype number 1. Now, just public clonotypes. So, the antibodies are made up of different genes, and one of them is the variable heavy chain gene, and that's paired with a light chain gene product. And so certain combinations are very common and are called public clonotypes. Right, Brianne, is that right?
Yeah, so basically the idea is that the B cells are using different strategies to potentially make an antibody. And a public clonotype is where multiple people's B cells all came up with the same strategy. It's sort of like a public strategy. So Rich's, Vincent's, and my B cells all use the same amino acids and same nucleic acids to kind of make that antibody. Whereas you could also imagine there might be a situation where one of my B cells came up with its totally own unique strategy to make that antibody, and that would be a private clonotype.
So they find 3 public clonotypes in most H2 naive individuals, right, after vaccination. But these are barely detectable in the people who are already exposed to H2.
And so that starts to make you think about the imprinting part, because the question is, are these individuals who were previously exposed just reusing their old response and kind of hoping it's close enough? Or are they making a new, unique kind of response to this new, unique antigen? Because it's not exactly the same antigen that they saw 50 years ago.
Right.
And so what we see is that a unique response to this antigen does look a little bit different. It involves these public clonotypes, which are not the same responses that the previously exposed individuals are making. So it's not as though we're making kind of a new de novo response that fits this antigen really well. We're just using the old one as and hoping it's close enough.
So I don't quite understand in a way how the public clonotype fits into this. I mean, because why? 50 years ago, was the public clonotype different?
So I think that to me, it's less about, I care less about whether it's a public clonotype and whether it's a private clonotype. And I care more about the fact that are the two groups making the same clonotypes or different ones? It just so happens that one group is making a public clonotype and the other group isn't.
Do we know what the other group is doing?
From here, I am not sure that I do.
Okay.
But I don't have a great answer.
Fine.
So they actually made a bunch of monoclonals
84 to be precise. My God.
From each of these 3 public clonotypes, just to see if they bind the same epitope, right? So they make the antibodies and then they look at binding, and they find that they're all binding the H2 head, right? The hemagglutinin has a globular head and a stalk attached to the membrane. So they're all binding the head, different parts of the head, but they're binding all the head. One side of the HA and the other side as well. So they concluded public clonotypes were elicited from the naive B cell pool in the H2 naive individuals, and they were not detected in the repertoire of H2 exposed individuals. So I also don't understand. So presumably when the H2 were exposed 50 years ago, they had these 3 public clonotypes, right?
Not necessarily. They made some clonotypes that were the best clonotypes at that point, but remember that they are being infected with the whole virus, and they're getting competition amongst B cells responding to all the antigens of the virus. And so perhaps the HA clonotypes were not the most important ones. So their immune system went a different direction.
Okay.
Oh, so they're gone.
And so maybe those are gone because we're only boosting with HA.
Right.
Or we're only looking at HA here.
So I don't know if this is the time in this to ask this, but I'm going to go ahead and do it anyway. Because you mentioned, Brianne, that the H2 that they're being vaccinated with is likely not exactly the same sequence or antigenicity as the H2 that they encountered initially. So the question becomes, if we're re-stimulating existing memory cells, what is the specificity of those things? Does it, I presume that, and this speaks to the whole issue of imprinting, I presume that those carry the antigen binding sites of their original encounter and not the new guy?
And so I would assume that those have the specificity for some antigen that is the same between the new one and the old one.
Ah, okay. Because they are getting stimulated.
They're getting stimulated.
So they're seeing something on the new antigen. Okay.
Yeah, so they're seeing perhaps part of the antigen that's conserved between the new and the old type.
Nevertheless, they have been trained differently initially. They're not trained entirely on the new antigen.
Well,
so it may be a little different in specificity.
Well, so I would think about it that there's a population of B cells that each can respond to different parts of the HA.
Okay.
Some of those parts are conserved. Some of those parts are new. And in the people who are getting exposed for the first time, they're going to, will pretend, make equal amounts of antibodies to each target, whether it's a conserved or not conserved part. Because as far as they're concerned, they're all new.
Okay. All right.
And in the people who were previously exposed, the memory cells are going to make a response to the parts that they've seen before, the conserved parts, and are going to sort of inhibit the naive cells that are seeing a different part for the first time. They're going to be like, sit down, Junior.
Did they actually do that?
There is some evidence that they do that.
Okay.
In other situations, there's evidence that they actually they inhibit the naive cells.
Okay.
Sort of because you have you have enough of a response already. You don't need to spend the resources making a new response too.
Right. Are those data in this paper?
No.
I didn't think so.
That is some general immunology that I'm thinking about there.
So they continue on the characterization of these antibodies. They make 84 distinct, and they don't include the public clonotypes here. And they look at affinity for binding the HA, and also whether they can neutralize drifted H2 HAs. So they find that the affinity of the monoclonals from people who were exposed versus naive, they're much higher in the exposed group, right? So They've undergone affinity maturation. That makes sense. Plus, these antibodies from people who were not exposed before vaccination to H2 cannot neutralize drifted H2 strains. So they have a 1967 strain, just a year before H2 disappeared. So it's drifted for 10 years. And only one monoclonal from the H2 naive group will neutralize that. And the others are much better at it. So the people who have seen H2 before, 50 years ago, right, they make antibodies with greater potency and cross-reactivity than B cells from a de novo vaccine response. But they also say that these H2-specific B cells in the naive individuals continue to affinity mature and gain potency after six months after the second vaccine
Yeah,
dose.
exactly.
6 months, yeah, which they can do in this kind of a study, right?
Yep.
They have to take time points. Let's see, what else is interesting here? So this is kind of interesting. They're comparing IgG and IgA antibodies that they've made here. And they noticed that fewer monoclonals in the H2 exposed people have, from the IgA compartment, have hemagglutination inhibition activity compared with IgG. And so they're thinking maybe those antibodies are targeting some other part of the HA other than the sialic acid binding part. And in fact, they do that. They look at that, and they see that these IgA antibodies are binding elsewhere. They're broader neutralizing antibodies targeting more conserved sites on the HA. So the IgA are broader, which is interesting because we kind of ignore them, right?
Oftentimes, yeah.
Yet they're doing better than the IgGs. All right. Then there's a whole long study of classifying these memory B cells according to surface markers. And, they put them in 7 classes or clusters.
Mm-hmm.
And in the end, they say we can find multiple clusters before and after vaccination. And that's it. We don't know what it means. But there are memory B cells there. Right. So unless there's something more than that, Brianne.
No, that's about right.
Sometimes as I read these papers, I think, okay, so they got this information. They're writing this paper. They need to report it. Okay, so it's recorded here. But I don't necessarily have to, especially if they're not going to slam me with it, attribute any special importance to it.
Yeah, it's in the literature, and other people might be able to do something with
Yeah.
it. As then they use these clusters to look at the B cells in the naive and the non-naive groups, and they say, we see different clusters in the different groups. We have more atypical B cells and a higher expression of memory B cell markers within the classical population. I think that's cluster 1 and 2 in the H2 exposed individuals, but we don't know what that means.
Yeah, I mean, I think that for me, what I take from this is first, they can come up with really fine distinctions of kind of B cells.
Yeah.
And then they can compare those special groups of B cells between the naive individuals and the previously exposed individuals.
Yeah.
And they say that, they look different. And so that might be further evidence that there's imprinting going on and that those previously exposed individuals are relying on the old types of B cells instead of making a new type.
Right. All right. So those are the data. So basically, if you've seen H2 50 years ago, you make a great memory response. You make high affinity antibodies. You make a lot of them, good quality, broadly neutralizing. And if you've not seen it before, then it takes you 2 doses to start to come close to that.
So I got one question that was burning for me through this whole thing. First of all, I'm not quite sure how close H1 and H2 are. I think in order to be different subtypes, you have to have less than 90% identity or something like that.
Mm,
So they're
yeah.
like 10% different at least, but that doesn't necessarily tell me how antigenically different they are. But I presume, I don't know for sure, that there is some, well, they say that they do talk about antibodies that they call H1H2 that are recognizing conserved epitopes.
Yeah.
So there are some, at least, epitopes on the H2 antigen that are cross-reactive with
Right,
H1.
right.
So my question is, they don't really talk about this, at least not that I can detect. In the population that are H2 naive, those dudes are imprinted on H1 or H3. So my question is, during the whole course of this, how do they respond relative to those memory cells? Okay? Does the H2 antigen stimulate some of that memory response? And if so, does that compromise the, as we were talking about previously, the response to the H2? This paper doesn't address that, right?
It does not address that.
Is it a
No.
legitimate question? Am
It
I
is a
thinking
legitimate
straight?
question.
Okay.
Yeah, it is.
I think that it's possible that an immunologist who knew much more about the immunodominance hierarchy, which is really what you're thinking about in influenza, might have more of an answer. I know that it has something to do with the immunodominance hierarchy.
Because to me, that's a component, a critical component of this whole imprinting thing. Okay, is if you're imprinted on something and you encounter something else, it may compromise. It may make it so that you don't have as slick a response to the something else as you would like, because you're
Yeah.
distracted with your original imprint.
Well, it depends how different it is, right?
Right.
Let's say you're born and as a kid you get immunized with H1 and H2 because that's what you're going to get, right? And then we have an H5 pandemic. It's probably different enough so that you're going to make a de novo response to
Could
H5.
be, yeah. Right.
I don't think, but among drifted, if you get a seasonal H1, H2, which is dealing with drift, it may just
Right.
elicit the original antibodies and it doesn't help you, right?
Yeah. So I mentioned a past paper that I remember reading about this that kind of talks about this, and so I'm putting that in the show notes right now because they were actually looking at what type of influenza was your first influenza and how they classified all of the hemagglutinins into group 1 and group 2. And they sort of said that you either, based on what your first influenza type was, it meant that you were either generally better against group 1 or group 2 and sort of impaired against the other.
Right.
So I put that in because that's what I was thinking about a lot when we were reading this.
So I was primed on H2, right? Yeah, 1953. No, H1.
Yeah.
In 1957, H2 arises, H1. So I'm presumably better off because H1 is still circulating, but not against H3. So it was when I got the two vaccines together, H1 and H3, I'm making a nice memory response to H1 and a de novo response to H3 the first time I got it, I guess. Right. Yeah, so the problem is that you're not going to find out what each person has got. You have to somehow get around this in a broader way. I don't know what that will be.
The part of the issue with all of this is influenza has lots of proteins, not just HA.
Right.
And it seems like we predominantly make antibodies to HA. And they imprint or our immune response, so that influences what happens later. And so perhaps we need to figure out how to really think about what's going on with immune responses outside of HA or outside of those main antigens, so we don't have imprinting. And so those other responses can protect us no matter which type of hemagglutinin we get infected with.
The other comment they make in this is that understanding all of this, and this was interesting to me, and almost I'm hesitant to say it, almost creepy. They say that understanding this is important because you may want to get to an infant before they're naturally exposed and give them something that will give them an imprint that will serve them better than the natural infection in the long run.
Good point.
Okay?
Yeah,
Good point.
absolutely.
We need to know what that is.
Exactly. And I think that's why I find it creepy. Because we don't know what that is.
Well, but what it could be is it could be
Hmm.
giving them an imprint against where their predominant memory response is against an antigen other than HA.
Okay.
So that when they're infected, they don't go in an immunologic direction of H1 versus H3, but they're making a stock response.
Yeah.
That's common.
Some conserved thing.
Yeah, exactly.
Yeah.
So we did a paper not too long ago on the benefits of antibodies to neuraminidase, right? They can also be protective. So maybe that's...
And they don't drift as much, right?
No, they don't drift as much. They do drift, but not as much. The other thing I wanted to mention was that, they say these people who are de novo, they're still doing somatic hypermutation 6 months after the boost. But can we say that the people who had been exposed 50 years ago, 50 years later, they're no longer doing somatic hypermutation? I think that's what the data say.
That's what the data say, yeah.
Okay, so that's our window. Between 6 months and 50 years. Somewhere in there it stops. Interesting stuff. Okay. Yeah, this is good. I like this.
Yeah.
We're doing a lot of immunology papers.
That's fine with me. I
Yay.
can't believe how much immunology I've learned on this show.
Well, I think it's practical for people, right? It relates to their susceptibility and so forth, whereas, understanding transcription, not so much.
My granddaughter recently had an exam in her medical microbiology course in high school on immunology. So she had all these immunology notes that she was going over. I really had to restrain myself from jumping in and immunologically confusing her. And then it's tough when you got like 20 minutes before dinner to talk about this kind of stuff, because my tendency is to go back to first principles and talk about what all this stuff means in a larger context. But you got to be careful, because for a kid studying for a test tomorrow that has a certain package of notes, that could do more harm than good. Could be confusing.
So the way I was taught to add columns of numbers is imprinted on me, because when I see kids nowadays using other methods, I go, it's no good. That's going to be imprinted on them. I can't use these new methods. Let's do a couple of emails.
Good. Been a while.
I'm going to take this first one about polio. Eric writes, "Thanks for all you do. I will miss Dixon's wit and his non-judgmental positive attitude. A polymath indeed." Yeah, Dixon was not judgmental at all. That's true.
That's great.
Yeah, that was true about him. Wanted to ask about a topic I remembered in a pediatric infectious disease discussion 30 years ago in med school. At the time, a comment was made about the idea of possibly distributing one IPV or even two in all children for hard-to-reach areas, then follow with OPV to finish the series, as it was more feasible to deliver. If you're going to do two IPV, and you're done. You don't need to do OPV. And there's no advantage to using OPV, and it's just going to reintroduce more OPV into the environment. Anyway, I'm sorry to interrupt. I remember in that discussion comments made that the vaccine-induced polio disease dramatically dropped after one IPV followed by OPV. Yes, it does, obviously. But there's no risk of VAP with just IPV alone. Is there any evidence for this? Yes, there were trials done where you can see a drop in VAP after giving one IPV followed by OPV. Is there any evidence for this? Could it be of any benefit or just another chance for OPV harm? If true, does adding GI tract involvement with OPV make a difference in immunity? With combination of OPV and after IPV in series, would there be any significant effect on GI shedding, risk of vaccine-derived polio, and risk to others from feces? Okay, so let's do some fact. I had to write this down so I remember it. about half of patients seroconvert after 1 dose of IPV. And it's 80% protective against the development of paralytic polio. 2 doses are 90% effective at preventing polio, and 3 doses are 95% effective. This is IPV. However, the recommendation of 1 dose of IPV followed by 1 of OPV is only partially based on these data. The other reason is that for 1 IPV followed by 1 OPV is to automatically protect the vaccinee from paralytic disease, right? That's the whole idea there. Additional doses of OPV do not harm, as Marina Chumikov gave her sons OPV every winter in order to confer nonspecific protection against severe disease following flu infection, so the kids would not get flu. And Chumikov talked about that on TWiV a while ago. While it was thought that OPV universally reduced virus shedding after vaccination or infection, it does not. For children under 1 year, OPV may reduce shedding, but as you age, OPV does not reduce shedding, and the possibility of the vaccinee shedding neuroinvasive virus after another dose is similar to that of an IPV vaccinee. Therefore, the risk of VAP to a secondary contact is the same. It's folklore to think that something you shed into the environment would prevent transmission. It's not well understood how IPV or OPV modulate the selective pressure of the gut used to restore fitness to the OPV-derived virus. So basically, when you get OPV, you can shed it for months. So how is it protecting your gut from infection if you're shedding it for months? It doesn't make any sense, right?
Now, I remember there's a component of this discussion that involves gut immunity. Is that right?
Yeah, but whatever it is, it doesn't help prevent shedding with OPV. So there's no gut immunity with IPV.
Right.
Perhaps there's some transient gut immunity with OPV.
But it doesn't do you any good.
You can still shed for months, right?
Well.
And you're immunocompetent, so it's not like you're immunocompromised in any way. So, in fact, people say OPV prevents transmission. There's no data that actually says that. It's folklore. All right. Hope that helps. Rich, can you take the next one?
Okay. How would I pronounce this? Raihan?
Yep.
Raihan writes, Dear TWiV team, episode 1195 was a perfect tribute to Dixon. That must have been when we were all here, right?
Yep.
Like many listeners, I too have had a career that was influenced by him. I remember listening to the first episodes in 2008 and hearing the two of you, that's Vincent and Dixon, bantering about science. So much has changed, but my admiration for both of you has nothing but grown. I was holding back my tears when you were all signing off. I could even hear Dixon chiming in when you said it's viral. Words can't capture what my heart wants to express. Just thank you. I am forever beholden to you. Best wishes, Raihan. Longtime listener, lapsed email writer, forever a fan. Isn't that nice? That's great. That's a nice tribute to Dixon as well.
Yeah, I was thinking, I started TWiV with Dixon, but then pretty quickly other people came in, right? So it was no longer one-on-one. And maybe that's what I was trying to recapture by starting TWiP, because that was the second podcast that we started, just to get back to me and Dixon gabbing, right?
Well, it's interesting that Raihan remembers listening to the first episodes in 2008.
Yes.
He'd been around for a while.
Yeah. Yeah, it was a lot of fun just to talk to him. Yeah. They're all good. I like TWiV. I love TWiV, but it's different, right?
Mm-hmm.
And plus, I didn't know anything about parasites, so it was fun to learn from Dixon. Brianne, can you take the next one?
Sure. Ida writes, Hi, I am an internist in the Boston area and an avid fan of TWiV. I appreciate your review of Tessandier et al.'s study on HPV dynamics, which sheds some intriguing light on an aspect of HPV that doesn't get much attention. I did want to clarify something on the clinical end, though. Current guidelines recommend that anyone with a cervix have Pap tests looking for cellular changes every 3 years from age 21 to 30. HPV testing with or without Pap testing is recommended every 5 years from age 30 to 65. More frequent testing does not seem to be valuable unless the person being screened has abnormal test results or has some other factor putting a suggestion for an especially high risk. The medical establishment has struggled for a long time with the question of whether the pelvic exam has a value apart from the PAP and HPV test. As far as we know, offering regular pelvic exams to women who are not having any local problems does not improve health outcomes in any way. PAPs may show yeast infections, and they may suggest the presence of bacterial vaginosis. But we do not treat either condition without symptoms, and if a woman has symptoms, we use other tests to make the diagnosis. In rare cases, a PAP may show uterine cells in a postmenopausal patient, and that finding could lead to an early diagnosis of uterine cancer. But this is akin to diagnosing lung cancer after doing an abdominal CT scan. You may occasionally save a life, but that doesn't make it a useful screening tool. Ida.
So
yeah, that's great. Yes, Rich.
There's something in here about basically HPV testing that does not involve a PAP test. Does that mean basically you're looking for sequence somehow?
Yeah, so you do a swab and a PCR. So when I was in Sweden last year, I did a TWiV with a doc who does this. And he said in Sweden, they stopped doing PAP tests because during COVID, you couldn't do a PAP. So they sent swabs to everyone, and they found that that was just as good. He said if there is, he said the only thing they worry about for cervical cancer is HPV. And if there is no HPV, that's it. End of story. They don't, but this doc shows there could be other things going on.
So would that would be a vaginal swab?
Yeah.
Yeah.
Okay. And so they can not only see whether it's HPV, but they can see the type.
Yeah.
Right. So they can tell. So if you've got high risk HPV, okay, that all makes sense.
Anyway, he says now in Sweden they don't do PAP smears anymore, but I don't know if there's exceptions like Ida. But it could be different from country to country as well, right?
Right. Well, and Ida also points out that it's different in age groups where they're looking really for those cellular changes earlier on. Yeah. And then after that, they're looking for the just the virus. And I would imagine that if you have cellular changes that early, that would be a particularly problematic indication.
Yeah. I mean, he said if you have a positive HPV PCR, then they will look to see if there's any cellular change, and then if there's a lesion, they'll take care of it, right?
Yeah. The thing that's really important about this is that if you get the diagnosis, you can cure it.
Yes.
Okay. You don't have to die of a cervical carcinoma. All right. But you got to get the diagnosis. And you got to get it right, and you got to get it at the right time.
If you're not screened and then you suddenly are bleeding and they look and there's a big lesion, then you're screwed, right?
Right.
So you have to do screening.
But also that, that it is possible to do a screening that is not just a PAP, that is not a PAP.
Yes.
Right.
Yes. So, I was just talking based on my conversation with him. It may be different here in the U.S., so I appreciate Ida.
Yeah, that's great.
I love so this gives me such confidence in medicine. Look, she's saying how you do things. There's a logical sequence of events that you follow, right? I like that. It's not just random. We'll try this, we'll try that. Everyone with a cervix, PAP tests every 3 years, 21 to 30. If this, if that. I just love that a lot.
This is there's this phrase, I believe I got the phrase right, that has always struck me as weird. Science-based medicine.
Yeah.
And I'm going, wait a minute. I thought medicine at least should be science. But no, a lot of it's empirical.
Yeah.
Right? And in a way, anecdotal.
And
right?
Yeah, I was going to say sort of more observation-based and not an anecdotal, as you say.
Yeah. It's funny you should say science-based because that's a website, right? Science-based medicine. It's my pick today. Anyway, let's do some picks of the week. Brianne, what do you have?
My pick is a site that came across my Blue Sky feed. And when I saw it, the first thing I thought of was this reminds me of things Dixon picks. Yeah. Or Dixon has picked. And so I sort of have this pick a bit in honor of Dixon. So this is coming from a group called The Node, which is a community of developmental and stem cell biologists. and their collaboration with another group called Focal Plane, which is a community of microscopists. And they have, this group has had a lot of images nominated for their best image in this image competition, and they have now chosen 15 images that are on the short list that people can vote for. And there also is a presentation of them in a gallery at a conference that's coming up next week in Liverpool. But I just really like looking at these 15 shortlisted images that are absolutely amazing and are well described. I sort of liked the descriptions with many of them. I can't decide which one is my favorite, though there are a few that I am rather partial to, but I thought that these were 15 amazing cell biology, developmental biology images that people might like to see.
They
beautiful.
are amazing.
Yeah, Dixon would like this.
Ooh.
It's looking at this, I just realized why the administration wants to destroy science in the U.S. because we have too much fun. We have more fun than anybody else. I mean, doing stuff like this, what could be better?
Like, look at number 4. Number 4 is an unknown organism. It's a view of an unknown species.
Yep.
How cool is that?
Number 2.
Number 2 is really pretty.
That's beautiful.
Really pretty.
And number 4 is like dark matter of biology.
Yeah.
We see it, but we don't know what it is. Yeah. Rich, what do you have for us?
Okay, this is chosen strictly for its nerd content. It is a publication in a Nature journal called Communications Engineering, entitled "Periodic Cooking of Eggs." So this is a total nerd flip out on how to cook an egg. Okay, and the deal is, as most people who have cooked eggs will realize, the white cooks at a different temperature or cooks differently than the yolk, and that is temperature dependent. And so, for example, when I fry, or in particular poach an egg, and I do a lot of this, okay, because I like cooking. If you're poaching an egg in particular, the challenge is to get the white done, okay, without overcooking the yolk, all right? And they point out here that they've got it all figured out as to the different cooking temperatures of the white and the yolk and how you're going to manage this. And this is so full of data and weird stuff. It's amazing. But if you go down all the way to figure 4, they show you a head-to-head comparison of all of these different methods that they look at for cooking an egg in a shell. Hard-boiled, soft-boiled. They got another method here that I can't pronounce. Sous vide.
Sous vide.
And periodic, which is their method, okay? And they say their method, periodic, is absolutely the way to go, okay? Because this really cooks them all the same. Well, the periodic method, it turns out, is you do 2 minutes in boiling water and then 2 minutes at 30 degrees, a bowl of water that's preheated to 30 degrees, and then back to the boiling water, and then back to the 30-degree water for a period of 32 minutes. So it's 8 cycles, okay? I'm not doing that.
Not happening. But also,
they did spectroscopy on their eggs.
Oh, yeah. They got the whole thing
all kinds
here.
of data here. All kinds of data. They measure the hardness. The problem is by the time you go 8 times back and forth, your 30-degree water is going to be cool.
Yeah, well, you're going to have to take care of that somehow.
But I want to know how you make. I want to know how you make poached eggs, Rich.
I make poached eggs?
Yes.
Okay. Well, first of all, I do put some vinegar in the water.
Okay.
I've gone back and forth about this, but I think it does help coagulate the white a little bit so that it doesn't sort of smear out all over the place. I have a big pan of water that is just barely simmering, and it's deeper than the eggs themselves, just barely simmering. So, if a few little micro bubbles coming up, it's probably at 210 Fahrenheit or 98 centigrade or something like that. And I just drop the eggs into. Actually, you're better cracking them into a little bowl and sort of pouring them in, so it's a little more gentle. And they do sink to the bottom, so I have to kind of get them off with a regular spatula. And I just watch them and I poke them with my fingers. That's the way I cook most stuff to tell whether it's done, is poke them with my fingers. And I have a big kind of a ladle. It's one of these perforated spoons that's used for cooking in a wok. All right. And I use that to ladle. You could use a regular holy spoon to ladle the eggs out. And I kind of, play with them. And it's not perfect, man. This is one of the few things that I don't time. Okay. I do this entirely. It's a spiritual experience. Okay. And that's it. I don't use a frame or anything like that. And, I get it maybe half the time.
As the students would say, you cook it based on vibes.
Yeah, there you go.
Yeah, so, all right, you put the egg in. You don't put it in a little coddler or something like that. Okay. Because I think I did the coddler thing last time I made a poached egg. But I did use vinegar, but that wouldn't make any sense with a coddler because it's not going to touch it.
Yeah. I mean, sort of the compromise situation is, in particular, if you're a person who occasionally eats tuna fish, is to save all the tuna cans and cut both ends out of them. So you have basically a ring that's maybe about 2 inches deep, and you can stick that in your boiling water and crack the egg into that. So that limits the spread to some extent. And so it works a little like a coddler, but not really. You're still poaching it.
So I remember I put vinegar in the water when I made hard-boiled eggs.
Ah, okay.
I found a recipe which says it's much better if you do, but it's another thing where you, I think you bring it to you put the egg in and then you bring it to a boil, something so it doesn't crack. Right? If you put an egg into hot water,
that's one of the methods in this paper, or that they refer to in this paper.
I see. But did they talk about how it tastes in the end? Probably
not. No, as a matter of fact, I don't think they did.
No, but they did do a principal component analysis of nutrients.
How'd they do that? They had to analyze it in some way.
Probably a mass spec or
they
something.
did NMR and mass spec.
I wonder who supported this research.
Oh, good question.
Who paid for it? Let's see.
Uh-oh.
Yeah, NMR.
We're going to get them in trouble.
Oh, I'm sure they're okay. Where was the work done, by the way?
Might get cut.
Fresh hen eggs were purchased at a local supermarket in Naples, Italy.
Ah, okay.
So they're beyond the fray. They don't have to worry.
Right.
Naples, Italy.
So see all the math and the methods?
Math. Oh my God. Interesting. They don't tell you who supported it. Let's see.
It's got to be somewhere.
Research was funded by the Italian Ministry for Universities and Research.
There you go.
In a project called 4D Printing of Smart Soft Robotics. I think
they were kind of straying outside their mandate there, I think.
Yeah. Well, I think I might make some poached eggs tomorrow by your method, Rich.
All right.
What do you put, a teaspoon of vinegar?
Oh, no, it's more than that. In a big pan, you can put as much as a quarter of a cup. You have to be a little careful because you can overdo it and you wind up with eggs that taste like vinegar. But the big thing for me really is lifting them out, on the perforated spoon, okay, and holding them over the water and poking the yolk with your finger, okay.
Yeah,
and kind of wiggling it around and seeing if you can get a feel for whether the white's actually done, but you don't want to. I mean, you got to. It's got to be a little bit firm on the yolk, so it's because if it's not firm enough, probably the white's not done yet. Okay, but I'll tell you, I'll bet you there's probably a 30-second or 60-second window where you've got the white cooked and the yolk's not overcooked.
Okay.
Hollandaise, man. Next episode, I'll tell you how to make hollandaise because this is it. Eggs Benedict, right? That's the whole point.
Right. Okay. My pick is not about food. Sorry. It is a blog post in a website called Science-Based Medicine, which I've been reading for years. But let me just tell you what science-based medicine is. It is it's dedicated from their writing to evaluating medical treatments and products of interest to the public in a scientific light and promoting the highest standards and tradition of science in healthcare. Online information about alternative medicine is overwhelmingly credulous and uncritical, and even mainstream media and some medical schools have bought into the hype and failed to ask the hard questions. We provide a much-needed alternative perspective, the scientific perspective.
Excellent.
Good science is the best and only way to determine which treatments and products are truly safe and effective. So these are all scientists and doctors who write, and I like it. It's very good. So the pick is an article called "The Use of Placebo Controls in Clinical Trials," and it's roughly divided into two sections. In the first, they tell you what a placebo is, and then they talk about the placebo effect, which they end up saying is BS.
Oh.
Right? They say it's BS. Let's see, where is this? They say the apparent overperformance of placebo groups in studies created the impression of a potent therapeutic effect. And they say this gives you a reputation as a mysterious phenomenon with great scope and power. And they say basically, if the placebo effect was a thing, we don't need to do clinical trials. We don't need to do placebo-controlled clinical trials. And they say we often try to demystify the placebo effect. So here's the bottom line for placebo effect. A variety of biases and statistical artifacts can conspire to create the illusion of a response to a placebo. When carefully controlling for these variables, the placebo effect virtually disappears for objective measures. What is an objective measure? A variable that the investigator can measure, like survival, blood sugar, temperature. The people who say they talk about pain and anxiety, they say, oh, I feel great. It's impossible to measure, right? And so they're saying you can't listen to that. You have to go by objective measures. And if you do that, there's no placebo effect.
Okay. I get it.
All right. So then they talk about designing clinical trials. And they say, there's a lot of bias that can come into design of trials, and a good investigator knows how that bias is going to happen and how to avoid it. Randomize it, why you want to randomize the subjects, why you want to blind the subjects. And this is very good. I'm going to read this to you. Blinding describes a study design in which knowledge of the treatment group is withheld from relevant individuals involved in the conduct of the study, including the subjects, right? Imagine that you're a participant in a clinical trial of an experimental drug. Now imagine that you're in the treatment group. You might be more enthusiastic about participating in the study. You might be more likely to comply with study activities. You might be more likely to report every headache, cough, sleepless night as a potential drug side effect. Now imagine you're in the control group. You might be more concerned that your condition is worsening. You may be more likely to access treatments outside the study protocol. You might be more likely to drop out of the study. You might be less likely to report life events as side effects. And that's what bias is. That's why we have to blind.
Yeah, I always
clinical
think about it
trials.
also with vaccine trials that if the two groups knew who had the vaccine and who didn't, there would probably be differences in how risky their behavior was.
Right. Anyway, then they talk about the value of a placebo control. So it's a great short article by David Weinberg, who is a vitreoretinal surgeon at the Medical College of Wisconsin. So these are always great columns. I like them very much.
Cool.
Placebo effect is BS. Now I have the ammunition to say it's BS. It's just self-reporting stuff. I feel great. Yeah, but your blood pressure hasn't changed, dude. Okay, listener pick from Anne. I heard an interview with Hank Green on the March 12th Public Health on Call podcast, a very informative podcast about issues affecting public health. Have either of you heard of that podcast?
I haven't.
I haven't either. He's written a book about tuberculosis called Everything is Tuberculosis, which I think deserves a TWiV or TWIM bump. Well, since we don't do picks on TWIM, I'm doing it here. And then they give some notes about John Green, or Hank Green, I guess.
So he's John Green, but his brother's Hank Green. They both are have some projects that they work on together.
Okay. John is not the one she's talking about here. She's talking about Hank. So they talk about John as being a New York Times bestselling author, a YouTuber. I don't know. She's got all this information about John.
Yeah, John is the one who wrote the book.
Oh, who's Hank?
Oh, wait. No, you're... So I think she has a typo because the Everything is Tuberculosis is by John Green.
Okay, I'm going to change Hank to John.
Yeah, that's by John Green.
Okay, so now they have some notes about John Green. And you can find him online. Okay, thanks, Ann. And folks, if you want to learn about TB, it's an interesting story, right? Name, Everything is Tuberculosis?
Yeah, I've seen the book around. I have not gotten a chance to pick it up yet because who knows when I would have a chance to read it. But I've heard good things.
Well, if we had the author on and you were on the program, you'd have to read it, right?
I would. I would. That would be fine because the last time we did that, I really liked the book.
Yeah, well, I will make sure only to pick good books.
Okay, good.
All right, that's TWiV 1203, 1203. It's amazing. It's 200 episodes since 1000, right? That's
I know. They're really adding up. It's incredible.
You can find the show notes at microbe.tv/TWiV. You can send your questions and comments and picks of the week to TWiV at microbe.tv. And please consider supporting our work. Go to microbe.tv/contribute. Brianne Barker is at Drew University. BioProf Barker on Blue Sky. Thank you, Brianne.
Thanks. This was great. I learned a lot.
Rich Conda is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Thank you very much. Always a good time. I really enjoy this.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.